begins with G1, which is a growing phase. The cell grows until it reaches a stopping point. If the cell is big and stable enough, then it will move on to S. But if it isn't, then it will stay in G1. Some cells stay in resting periods known as G0. During G1, proteins, carbohydrates, and lipids are formed. Proteins are synthesized using the endomembrane system. The endomembrane system begins after RNA messages are translated into polypeptide chains on ribosome. These protein units are then sent through the rough ER, where they are folded into a 3D conformation and where sometimes carbohydrates or sugars are added. The protein then leaves the rough ER in a transport vesicle to enter the cis phase of the Golgi apparatus. Inside the Golgi apparatus, further modification of the protein occurs. Finally, the protein leaves through the transphase in a vesicle to go outside of the cell, or in our case, the plasma membrane of the cell. After G1 comes S, which stands for synthesis. In this phase, DNA is replicated. In order for two similar daughter cells to be produced, the DNA instructions must be duplicated. If the DNA does not replicate, then the cell stays in synthesis. In DNA replication, a parent DNA molecule has two complementary strands of base paired nucleotides. Replication starts when the DNA is fed through a factory, which comprises of replication proteins clustered together. First, helicase unwinds and unzips the DNA into single strands. The point where DNA is separated into single strands is known as a replication fork. Next, SSBs, or single-strand binding proteins, coat the single strands to prevent them from merging back together. Before the DNA actually replicates, a good thing to remember is that the DNA strands are anti-parallel, meaning that the DNA strands run from a 5' to 3' in opposite directions. Polymerase enzymes only synthesize nucleic acids in this 5' to 3' direction. Since DNA polymerase can only extend a nucleic acid chain, to give the DNA polymerase a place to begin replication, an RNA polymerase called primase synthesizes RNA primers needed to start strand replication. DNA polymerase can now begin to synthesize a new complementary DNA strand. Two polymerase enzymes, with the aid of a sliding clamp, which helps hold the DNA polymerase onto the DNA, start copying DNA in opposite directions. One polymerase can remain on the DNA and copy it in one continuous strand. This strand is called the leading strand. The other strand can only copy a short stretch of DNA before running into primer of a previously sequenced fragment. Therefore, the polymerase must move back and begin copying from the extension of another RNA primer. The short stretches of DNA that make up a lagging strand are called Okazaki fragments. To remove the RNA primer so that the Okazaki fragments can be joined together, RNase H removes the RNA primers that previously began DNA strand synthesis. DNA polymerase then fills these gaps left by RNase H. Lastly, the Okazaki fragments are joined together by DNA ligase. The DNA double helix is reformed and replication is finally finished. Replication is called semi-conservative because each daughter DNA consists of half of a parental DNA and half of a new DNA. Once S is finished, G2, or a growth stage, occurs. Growth refers not to the growth of the cell, but rather a mass production of mitotic apparatus, or materials used in mitosis. Examples include spindle fibers, microtubules, or centrioles. In our cell model, the centrioles are being mass-produced. Once G2 is completed, mitosis can finally begin. For mitosis, we are focusing on the organelles in the cell that are used specifically in mitosis. Mitosis starts out with prophase. Prophase is considered the longest phase in mitosis. During prophase, the centrioles migrate to the poles of the cell. Spindle fibers form from microtubules condensing, and chromatin condenses into sister chromatids. Lastly, the nuclear envelope breaks down. After prophase comes metaphase. During metaphase, sister chromatids line up at the equator of the cell, forming what is known as the metaphase plate. The spindle fibers run from the centrosomes to the centromeres of the chromosomes. After metaphase comes anaphase. Anaphase is considered the shortest phase of mitosis. During anaphase, karyokinesis, or the splitting of the genetic material, occurs. The spindle fibers shorten, which pulls the sister chromatids apart. The cell also elongates due to actin and myosin. Telophase is the last step of mitosis. During telophase, the spindle fibers recondense into microtubules. The nuclear envelope reforms. The sister chromosomes decondense into chromatin and the centrioles migrate back to their original positions. After mitosis, cytokinesis occurs. Cytokinesis is when the cytoplasm divides and is involved in the splitting of the parent cell into two new daughter cells. During cytokinesis, actin and myosin pull closer to the equator of the cell, which forms a cleavage furrow. This constriction goes on until eventually it slices into the center of the cell and the cell divides into two new daughter cells.